attendance. All right, we are recording. All right, Bangura is not here. Casebolt is here. Trisden is not here. Covington Griggs is not here. Dow is here. Hughes is not here. Lebdy is not here. Little is not here. Mankins is not here. Mathis is here. Rains is not here. Reed is not here. Elise is not here. Thompson is not here. Yindui is not here. And Elkins is not here. So that's everybody who's in person. As usual, if you're online, you'll just need to send those words in before 9 a.m. And as usual, at the very end, the last word is going to be me asking you to ask me a question about the material presented. So make sure you get that. Think about that as I go through the lecture. And of course, if you're watching the video as opposed to live streaming right now, so if you're watching the video at a later date, make sure you take a screenshot every time I say one of the attendance words or phrases. And the first word for attendance is going to be coffee. Coffee is the first word for attendance. So let's jump into this. We won't do announcements or reminders yet. We'll jump back into where we left off. And where we left off was talking about this. Oops. Let me back up here. Sorry. Come on. There we go. Yeah, that's what we left off talking about. All right. So like we mentioned when we stopped talking about this last or on Monday, eukaryotes are a little bit more complicated than prokaryotes, right? So we humans, for example, and plants and protists, and fungi, we're more complicated than bacteria. Therefore, our process of translation, which this comes after transcription, right? So translation is going from RNA to protein, right? That's more complicated for us. And one of the things we mentioned, one of the ways it's more complicated, is this RNA that we just made in that process of transcription, it's not ready to go, right? So it's not like it's just done. As soon as we make the RNA, we can change it into protein right away, right? It has to go through some changes before it can even leave the nucleus. And one of the changes we talked about is cap and tail. We already talked about that, so I won't go into that anymore. But the other one, and I don't think we talked about this one. No, we didn't. The other one is RNA splicing. So for the exam, we need to know those are the two things that the RNA has to go through before it can leave the nucleus. You also need to know the difference between the two, right? So at cap and tail is exactly what it sounds like. We just add some extra nucleotides at both ends of the RNA strand. RNA splicing is also exactly what it sounds like. This is splicing parts of the RNA out of the molecule, or as the textbook says, it removes non-coding nucleotides from those that do code for amino acids. So remember, that's the whole purpose here. These words that we talk about when we talk about the genetic language, these A's and T's and C's and G's and U's, right? Those make up words, and each one of those words says, bring me this amino acid or bring me that amino acid. But some of them, you could almost call it junk DNA. I think probably for our course, we could call it junk DNA. As far as we're concerned in this class, they don't have any function. But if you want to look into it for independent work, some of it does serve a function, and we don't generally call it junk DNA anymore, but we used to. But what you do need to know is the difference between the two. So the non-coding regions, the stuff I'm saying you maybe could call junk DNA, those are called the introns. So the introns are the parts that are getting removed. The other parts that stay, the useful stuff, those are called the exons. That's the part that does not get removed. And that used to confuse me as a student, as a young student. Well, I wasn't young, but as an early student when I was a freshman. The word introns sounded to me like that's what you left in. And exons sounded like something you would get rid of, right? It would exit. So it was hard for me to memorize it for a while. So the way you need to remember it is the exons, that's the part that's getting expressed. Right, so the code in those exons is going to be expressed as a protein. The introns is the in-between garbage. And again, It does have a purpose, but for this course, we're fine calling it garbage, useless stuff, right? So remember, exons are the parts that are expressed, and the introns are the in-between stuff that we don't need. So you can see this picture down here. I don't think this is from your textbook, but you can see at the very top, right? We have that pre-mRNA. So it has the cap and the tail, which is not labeled the way you would normally expect it to see it, but that's okay. Anyway, it has the cap and the tail, but it hasn't gone through splicing yet, right? Because it has all those so-called useless introns, all those purple spots. So it goes through splicing, right? We remove this intron and then bring these together. We remove these introns and bring these together, and then there you have it. Then at the bottom, we have this new ready to go molecule of mRNA. Exons and introns are transcribed, right? The whole thing is transcribed from DNA to RNA. So we'll back up, right? So we've already talked about transcription. But now that you know about RNA splicing, I just want to back up and remind you or let you know that even though these introns are not needed, they're still written in the codes of the DNA, and they're still transcribed into the codes of the RNA. Otherwise, we wouldn't have a need to splice it, right? So this is something you need to know. Some things you could think of for independent work. For example, why do we even have introns? If they're going to be removed anyway, why are they there? Also, another thing, notice when we talked about DNA replication, I said, oh, the enzyme that does that is DNA polymerase. When we talked about transcription, I said the enzyme that does that is called RNA polymerase. Notice I haven't told you anything about what does this job. It's not, I don't believe it's in your textbook, and I'm not going to ask you. So it's just another thing you could look up for independent work. What is responsible? You know, which enzymes are responsible for cutting out these introns? And then bringing this other stuff back together. So think about it. That's got to be pretty complicated. Cutting parts out and then splicing the new part, the other parts back together, creating those covalent bonds. Anyway, any questions so far? There's a little bit more about RNA splicing, but are there questions so far? Okay. RNA splicing plays a significant role in humans because when you think about this, humans have about 20,000 genes, but we have many more polypeptides than that, many more proteins. And that should be, again, if you're thinking about things and you're a thinker, at first that should confuse you a little bit. Like, wait a minute. Genes are the instructions on how to build proteins. And without those genes, we can't have those proteins. That's the only way our body makes proteins is by reading genes, and transcribing it and then translating it. So how is it that we have 21,000 genes, but we have more proteins, right? That should make you think like that's not right. Actually, I'll get back to you. I'll explain that in a second. Looks like my slides are a little bit out of order. So keep that thought in mind. Here's just a visualization of what we said previously. So again, we have this DNA strand, exons and introns included, the whole thing, right? We transcribe it in the process of transcription, right? We add the cap, we add the tail, we get rid of those introns and splice the rest of it together. Then we're left with this mRNA that's now ready to leave the nucleus and go get translated into a molecule of protein. Now let me back it up. So what I was, again, going back to what I was saying earlier, we have about 21,000 genes, and that's something else you could look up for in independent work. Exactly how many genes do they think we have now? And also, how many proteins do we create? Things you could look up, because I'm not going to ask you on the exam. But 21,000 genes, we have more proteins than that. How does that happen? And it's this stuff right here. And this is called alternative splicing. I'm probably not going to ask you any questions about it, but it's worth mentioning. This picture is not from your textbook, but you can see here, again, we have this DNA strand that has an exon with an intron in between it. And then another exon, another intron, another exon, another intron, another exon, another intron. What I love about this picture is that you can see these exons in this example are all different sizes. Some of them are bigger, some of them are smaller. Same with the introns. The introns are also different sizes. It's a better representation than that last simplified picture I showed you where they were all exactly the same. So yeah, they are different. So then anyway, we have this molecule, right? We have this gene of DNA. We transcribe it into this pre-mRNA. We put the cap and the tail. Now we're ready to do some splicing. But now I'm telling you there's something called alternative splicing. So splicing is not always as easy as just removing every single one of the introns. Sometimes you remove some of the exons. So let's look at some examples. In this one on the left, this is just normal, right? So in this one, we just got rid of all the introns. They were in between all the exons, right? So we have all the exons here, exon 1, 2, 3, 4, and 5. And with that, whatever code that is, if you translate that, that's the protein you get. Here, we have exons 1, 2, 4, and 5, right? So here, when we got rid of all the introns, we also got rid of exon number 3. And because of that, it's technically a different gene. And because of that, you are creating a slightly different protein. Same thing here in this example. We have exon 1, 2, 3, and 5, right? So in this case, not only did we remove all the introns, but we also removed exon 4. And because of that, now you have a different code. So therefore, you have a different gene. Therefore, you have a different protein. Now, those pictures probably make sense to people who are biology majors and been doing this for a while. Those pictures at the bottom. But they don't need to make sense to you. The point here is that you can see whatever those weird looking pictures are, you can see that they're slightly different, right? And that's what this is all about. It's all about being that slightly different shape, which makes it a completely different protein with a completely different job. All right, so that's it for RNA splicing. That's actually it for cap and tail and RNA splicing. So at this point, we're done talking about transcription. So are there any questions about it? All right, let's do some announcements and reminders. Just so you guys know, my kids are on spring break. So I'm working from home. Well, obviously not right now, but most of the time I'm working from home and dealing with my 3 kids, which is really hard. And on top of that, yesterday I wasn't feeling very well. So put the 2 together. I barely got any work done yesterday. Didn't get as much done as I usually do on Monday. So I'm a little bit behind. Just wanted to let you know. Which brings me to this, and I'll jump ahead a bullet point. Someone already asked this question, and this is a good time, a good segue into this. So if you've already turned your lab in, I probably just haven't had a chance to look at it yet. This week's lab is virtual. You're going to need a computer and you're going to need headphones, but we're still meeting in person. That being said, when we're done with this class, I'm going to go and check right now, as soon as I can, and look at everybody's lab who has turned it in. I probably won't have time to grade it yet, but I can at least look it over and say, yeah, that's fine. You don't need to come to lab. So, yeah. So if you've already done it, like I said, I'm going to look at my emails. I'm going to take a look at them. And so if you have already done it, there's a very good chance I'll tell you specifically. You'll know. There won't be no question about it. You'll know that you don't have to come in. So any questions about that? All right. In next week's lab, same situation. It's virtual, but we are meeting. And next week's lab, to be honest with you, even though it's virtual, it is a little bit more complicated. So it might help you. Next week lab, even though it's virtual, what I plan to do is I'm going to do the virtual lab. Like I'm going to present it right here, project it so you can see it. That way, even though it's virtual, you all won't be doing it if you don't want to. We can do it all as a team. I'll set up all the parameters and run the simulation, and we can see the thing work, and then we can see the data, and then we can collect the data together. So again, next week's lab, still virtual, still need to show up, but I might be able to help you with it next week. This week's lab, even though it's virtual and we're meeting, it is very individual, as you guys have already seen. So any questions about labs in general? All right. Next thing, this is actually a reminder or an announcement, not a reminder. I will give you extra credit for proving that you're registered. So not just a little sheet that shows you got advised, but I need some sort of proof that you got registered, like maybe a screenshot of your MyState, something like that. For the next semester. So on that note, if some of you are about to graduate, then I'll also give you extra credit because you can't register if you're about to graduate. But if you show proof that you've applied for graduation, so applying for graduation or registering, if you do that anytime between now and the end of the semester, I will give you extra credit. I'll think about how much. I'll do it like independent work where I tried to bribe you. So I don't know the exact details yet, but the sooner you do it, the more points I'll give you. How about that? Anyway, any questions about that? All right. As a reminder, you can still retake exam 2. It's still open, unlimited, open book. Just let me know when you're done so I can adjust your grade. Again, a reminder, if you're in here in person and on time, you get 4 points of extra credit. If you're watching the video or joining us online, you get 1 point of extra credit. Speaking of which, to remind you, if you came in late, make sure you email me and say, hey, I came in late. It's better to get some points than no points. And then finally, again, the Monday after Easter and the Friday before Easter, we will not physically meet in this room. We will do office hours and lecture 100% online. So any questions about announcements and reminders? Okay. Next, like I said, we're done talking about transcription. So now we're going to actually talk about translation. But before we do that, and I like, again, this is slightly different than the textbook you're using. I'm basing this off of the old textbook. I like what your old textbook did because in this case, translation is a little bit more complicated than transcription. So I like that this textbook kind of, before we tell you how translation works, we talk about all the parts that are involved and explain what they do, which we didn't need to do for transcription, right? Only parts that were involved was the RNA polymerase, right? I mean, granted, there was other parts of the DNA that you needed to know, like the start codon, or excuse me, the promoter and the terminator. But there was only one enzyme that we talked about. So here it's a little bit more complicated. Let's jump into it. The next word for attendance, I'm not going to say it. I'm just going to circle it. So that is the next word for attendance. And of course, again, if you're watching the video, so not if you're live streaming with us right now, but if you're watching the video, I'm going to need a screenshot that shows that. Anyway, so let's talk about the parts. The first important ingredient is the mRNA itself. Obviously, this is what we've been talking about, right? This is the code, right? After that, we also need the machine that actually does the translating, which I'll talk to you here in a second. It also requires different enzymes and energy, right? Because if we're building molecules, that requires energy. That usually comes in the form of ATP. There are other sources. You don't need to know about them, but if you want to look that up for independent work, you can. So that might be the first time I've ever said that this semester. So far, the only thing we've ever talked about is ATP, right? When we talked about respiration, we talked about breaking down glucose and other molecules, right? So we could make ATP. But now I'm telling you, there's actually other little molecules that our cells use for energy, and you can look that up if you want. Anyway, translation requires two other components. Actually, I need to fix this slide, but it is true. It requires ribosomes, which is this. These two bullet points are the same thing. The machine that does the translating, those are called ribosomes. And it also requires yet another type of RNA, which is tRNA. So far now, I've told you about two different types of RNA, and you need to know the difference between the two of them. There's the mRNA, which we've been talking about. That's the message, right? Messenger, because it has the message. It says this, bringing the message that says, we're building a protein. First thing I'm going to need is this amino acid. Next thing I'm going to need is that amino acid, right? It has the message. The tRNA, as you're going to learn later, so this is still just I'll just do it. The tRNA is transferring stuff. So the question is, what is it transferring? And this is what you're going to need to know. Again, this tRNA, it's going to require an interpreter because, again, we're going from one language to the other, right? We're doing translation. So if you think about it in those terms, yeah, we need an interpreter, which we'll talk about here soon. As a reminder, we have three-letter words, right? So this language, this nucleic acid language, right? They only have 4 letters, right? So if it's DNA, A, T, C, and G. If it's RNA, A, U, C, and G, right? Only 4 letters in that alphabet, unlike ours. And in that language, all the words are 3 letters long. So as you know, as you've done, as I showed you previously, that means there's 64 different words. But anyway, this is where the tRNA comes in. tRNA matches the amino acids to their codons to form the polypeptide. In other words, remember, the message is in the mRNA, right? It has the code. tRNA reads that code and it transfers the amino acid, right? Because again, to remind you, the mRNA is just a bunch of words, and those words say, we're going to build a protein. First, I need this amino acid. Then I need that amino acid. Then I need this amino acid, right? That's what's happening. And the transfer RNA is the thing that's saying, oh, I got you. You need that amino acid? I'll bring it. Oh, you need this other amino acid? I'm on it. So that's how that works. Remember, the mRNA is just the code. If we were to use an analogy, this is slightly silly, but it was and I haven't introduced you to the ribosome yet, but imagine I'm the ribosome. I'm the thing that's building the protein. I've got my mRNA right here, right? It's been written down, so I'm reading the code. And you guys are tRNAs. and each one of you has a specific amino acid. And I say, okay, guys, first I need leucine. And then you're like, oh, that's me. I'm the one with that amino acid. And you come and you bring it to me, and I put it right here. And then say, all right, next I need proline. And then one of you says, oh, that's me. That's my amino acid. And then you come over here because you're the transfer RNA, you're the tRNA, and you transfer that amino acid to me, and I attach it to that first amino acid. And then I read my code again and say, hey, guys, I need tryptophan. And then someone else, you're like, oh, that's me. That's my amino acid. And you come down and you bring me the amino acid, and I take it and I attach it to the other amino acids. That's how that works, right? I'm the ribosome. I'm the thing that's actually putting those amino acids together. I'm the one who's reading. Well, I guess that's a kind of a bad analogy, but I'm the one who has the code, and then you, as the transfer RNAs, you have your specific amino acids, and when it comes time for you to bring it, you bring it to the ribosome, and then the ribosome squishes them together. Anyway, that was just an analogy. As we go through these slides, you'll get to see that in writing and in a proper explanation, not an analogy. Anyway, yeah, so again, you need to know the difference between mRNA and tRNA. tRNA is transferring those amino acids. It carries two, and to do that, it carries two essential functions. The first one is to pick up the appropriate amino acid. And again, like I said, using that analogy, every transfer RNA has a specific amino acid. So it picks up the correct one. And then again, it recognizes the code on the mRNA. So, yep, that's me. I'm the one, that's my code. I'm bringing this specific amino acid because the message says to bring this specific amino acid. And it's its unique structure that enables it to do both of those things. So I'm going to talk about the structure, but I'm not going to ask you questions about the structure. I just want to show you what it looks like to help you understand. Any questions so far? Again, we're still talking about tRNA. We're not done with that conversation yet, but before I go to the next slide, do we have any questions? All right. So there's what it looks like. You can see it's made up of a single RNA strand, right? This is one single RNA strand. You can see, though, that it has kind of looped up on itself. So some people might call that, if you didn't know any better, you could call that double-stranded, because you'd be like, look, there's hydrogen bonds between those. They're stuck together, just like two DNA molecules. But this is different. This happens to be just a single strand that's kind of looped up on itself. But these looped up on itself portions are important because, again, like I said, it's the shape that we're talking about of this tRNA, and it's the shape that allows it to do its job. This picture is not from your textbook, so you don't need to know about this loop or that loop or the acceptor stem or the variable loop. I might mention I'll talk about the anticodon later, but I probably won't ask you any questions about it. You don't need to know 5' or 3' or know that there's an OH here. This is so. Again, I'm probably not going to ask you any questions about the shape. I just wanted to point that out because that is a common theme in biology, right? Form and function. We're still not done talking about tRNA, but are there any questions so far before I move to the next slide? All right, as you can see in this picture, on one end of that tRNA is a triplet of bases, meaning there's three bases, right? And that is what we call the anticodon. And it is complementary to the codon and the mRNA. And in a sense, I haven't really told you anything new. I just gave you I gave you the word. The word's new. But I've already described this. Remember, like I said, tRNA, each tRNA has its very specific amino acid that it carries. And because of that, it also has a very specific anticodon. And the anticodon reads and recognizes its counterpart in the mRNA. So that's how the tRNA knows, oh, that's me. I need to bring my particular amino acid because that's my codon that matches my anticodon. So for the exam, I'm probably not going to ask you what an anticodon is. Almost definitely not going to ask you. But there might be questions that use that word, right? So as a question's describing some kind of scenario, obviously it's going to need to use the word anticodon if we're talking about a tRNA coming in and recognizing its code. Anyway, that's at one end of the tRNA. At the other end, obviously this should make sense because this is the whole purpose of this RNA, is to bring in the amino acid, right? So on the other end of the tRNA, is where the amino acid attaches. Here's a picture of what I'm talking about. These are two different drawings that show two different ways of showing the same thing. I like the one on the right. It's more simplified. This one's more realistic, that's showing all the twisting and the folding and the hydrogen bonds between the bases. And it shows the anticodon. It shows where the amino acid attaches. But this, I think, is better for 100 level class, right? All right, there's the tRNA. We'll do the part in the middle. Then there's the business end where the amino acid is, and the anticodon where it reads its own code, its complementary code in the mRNA. So yeah, that's what tRNA is. tRNA transfers the amino acids. And notice too, I haven't told you how that happens either. More stuff you can look up for independent work. How does the amino acid get attached to the tRNA? Is there an enzyme for it? If so, is there like one enzyme for each of the 20 amino acids, or is it like a one-size-fits-all kind of situation? We're like, yep, this is the enzyme that's always going to add amino acids to the tRNA. These are all things you can look up for independent work. All right. And that's the last part for the discussion of tRNA. Next, we're going to talk about the other parts of the process. Are there any questions at all about tRNA? All right. Like I've already mentioned earlier, using that analogy I was using, ribosomes are the thing that's doing the building of the protein, right? So the mRNA is the code that says, hey, bring me this amino acid, bring me that amino acid. The tRNA that we just got talked about is the one that brings the amino acid. Like, okay, you want this amino acid? Here you go. Ribosomes are the organelle that brings all this stuff together. So it's the ribosome that brings the tRNA to the mRNA and puts them together. And then, again, obviously that tRNA is bringing amino acids. So it's the ribosome that's attaching this amino acid to that one, and then to the next one, and then to the next one, right? We're just adding on to the chain of amino acids. Probably not going to ask you this, but just so you know, technically, a ribosome is made up of two subunits. Big old question mark. Is it like I might ask you, I might not. Those two subunits are made up of proteins, and yet a third type of RNA that you need to know now is rRNA, which is ribosomal RNA. So now there's three types of RNA that you have to know about. And just so you know, that's it. There are other types of RNA that you can look up for independent work that have much different jobs. But yeah, those are the only ones you need to know about. mRNA, which carries the message. tRNA, which transfers the amino acid to the ribosomes. And ribosomal RNA, which is what ribosomes are made of, that and proteins. So, again, these ribosomes are made up of two subunits. One of them is small, one of them is big. I'm definitely not going to ask you to differentiate between the small and the large subunits. But yeah, some of them are bigger than the other. It just so happens that the small subunit is where the mRNA attaches, and the large subunit is where the tRNA attaches. But again, I'm not going to make you, I'm for sure not going to ask you to differentiate which one goes with which. Of course, after seeing the picture a few times, it'll probably make sense, and you probably remember it anyway, but here's what it looks like. Actually, there's different ways. This is from your textbook, if I remember correctly, that or the old textbook. But I want to point out this is a very simplified version. And if you were to look this up somewhere else, you might find a picture of a ribosome that has not an A site and a P site, but also some third site. So yes, I would suggest just sticking with the textbook. Otherwise, if you're going to be looking at other things, and it might be confusing. I'm not going to ask you to differentiate between the A site and the P site. Just know that in the RNA itself, the ribosomal RNA, the ribosome has two spots, right? And yeah, one's called an A site, one's called a P site. It doesn't matter, but there's two spots for the tRNA. This part up here, that's the large subunit. The small part down here is obviously the small subunit. And again, like I said, hopefully after you see this picture a few times, even though I'm not going to ask you to differentiate between the two, you can kind of see it, right? The small subunit, that's perfect to have a strand of mRNA, like you can see on the right side. This large subunit is going to be large because that's where that tRNA fits in, right? It fits in like a glove. So yeah. And then you can see here on the right-hand side, we've obviously, the process has been happening for a while. We have two tRNAs in here. We have this long growing strand of amino acids. We have this amino acid that this tRNA just brought in that hasn't been added to the chain yet, but it's going to. And we'll talk about that process here in a second. Speaking of which, We're now done talking about the parts, so we can finally go on to talk about the process. Are there any questions about the parts that take place in translation? And again, the parts are all right there, right? We've got the RNA, we have the ribosome, that's the big blue thing. We have the mRNA, which is bringing the message. It says, bring me this amino acid, bring me that amino acid. We have the tRNA that says, oh, yeah, that's mine. That's my code. I'm going to bring in my amino acid. And again, those are the amino acids. So all the parts are there. Any questions then before we talk about the process of translation? All right. Process of translation. In one sense, this is kind of nice and easy because remember, when we talked about transcription, I said transcription itself is broken down into 3 stages, and you have to know them. And now I'm telling you the same thing for translation. But the good news is, at least it's the same words. Initiation, elongation, and termination. So here we go. The first one, initiation. This is basically, initiation is just bringing all the parts together, right? So we've got that mRNA that's already been processed, right? It has this cap in its tail, which helps it recognize the ribosome. It attaches to the small subunit. Then comes the first tRNA that has its very specific amino acid, and all the parts come together. And I'm going to show you a picture of that, but before I do, are there any questions about the words that are on the screen? All right, there, again, this is what the mRNA looks like. Again, you can see the cap and the tail. That helps it, the ribosome recognize it. So again, like I said, in initiation, all the parts are coming together. Technically, what happens first is the mRNA attaches to the small subunit. Then the tRNA comes in because it recognizes its code. Then the large subunit shows up. But what I'm definitely not going to ask you, so I'll put a big ol' X to this. You need to know those things happen, but it's a 100 level class for non-biology majors, so you don't need to know that it happens in that order. Like, it's not like you need to know, all right, first the mRNA attaches to the subunit, the small subunit before the tRNA comes in, right? I'm not going to ask you that. Just know initiation is when all the parts come together. And here, I like this. So you can see here, this picture is not from your textbook, because like I said, some spots, some pictures that you see will have a third spot in the drawing, and you don't need to worry about that. Also, a difference in this picture that you won't see in your textbook is, like I said, ATP is not the only form of energy. In this case, it's showing GTP. So remember, ATP is just a adenosine triphosphate, right? So it's a base, like a DNA base A, same A, except it has a phosphate group on the end of it. Here, same thing. GTP. It's just another, it's G, just like the DNA nucleotide G, except it has a bunch of phosphate groups on it. Anyway, let me show you the picture that actually came from your textbook, I think. If it's not your textbook, it's the one previous, but definitely from a textbook meant for a 100 level course. But again, here we go. We see the mRNA is attached to the small subunit. The tRNA recognizes its code. It comes in and attaches. It brings its amino acid, right? And then once all that happens, then finally the small subunit comes in and attaches. And not that it matters, not that I'm going to ask you this, I will point it out. But notice too, once this happens, that first tRNA is in the P site. Like I said, you don't have to remember, but it's in this one on this side, right? And the one on that side is open, right? It's vacant, if you will. Another thing to point out, and your book might mention this, so I'm going to tell you now, I'm not going to ask you to remember this, but they definitely showed it in the picture. But the first amino acid is always that, NET. You need to know that. But again, your textbook is going to say it. So if you're studying your textbook and you're reading that, like, oh man, do I need to remember that for the exam? No, you don't need to remember that for the exam. But you do need to remember it for the next attendance word. So the first amino acid is always what? The 3-letter word. It's on the screen. Anyway, any questions about initiation? And again, this is what we're talking about right now, is this initiation of translation. All the parts have come together. If you download the PowerPoint, you can watch this little video, this little animation that shows some things happening. So if that's initiation, let's talk about elongation. And again, it's in the name. I'm hoping this is nice and easy. What are we elongating? We're elongating that polypeptide, that chain of amino acids. We're making it longer and longer by adding amino acids to it. Here's where it gets a little bit more complicated. Elongation itself is broken down into 3 steps, right? So it wasn't like that for transcription, right? From transcription, it was just like, all right, we're going to have this nucleotide. Next is this nucleotide. Next is this nucleotide, right? We're just building a strand of mRNA. Here, it's a little bit more complicated, and we're going to talk about it. So, and I might ask you this or I might not, I don't know yet on the exam, but here's the 3 steps of the elongation itself. Step 1 is when the codon, it's called codon recognition, and it is in the name. That's when the anti-codon that's on that tRNA pairs with the mRNA codon. In other words, this is another way of saying step 1 is that particular tRNA says, oh, that's me. I'm coming in and I'm bringing my amino acid with me. And like I said, I'm not going to ask you this, but I will point it out. You can see it again. This is the picture we already saw previously. This was initiation, right? You can see this. This tRNA is right here in that spot on the left. The spot on the right is vacant. It's like, all right, come on in. And it does. The tRNA says, that's my code. I'm coming in and I'm bringing my specific amino acid with me. That's step 1. So let's see if you can figure this out before I go to the next one. Think about what the purpose here is, right? This is elongation. We're trying to make a strand of amino acids longer and longer. So far what I've showed you, and imagine in your head this is actually happening. This tRNA is coming in with the next amino acid. So in your brain, picture that it's there. Right here, the picture it's showing that it's going there. So in your brain, picture that it got there. So what do you think the next step is going to be? Because step 1, that tRNA says, oh, that's me. And it came in and it brought its amino acid. So again, in your brain, try to picture this tRNA sitting right here with its amino acid, right next to this tRNA with that long strand of amino acids. What do you think must happen in the next step? And it is kind of intuitive. It's very intuitive. Well, I'll ask you this. This is elongation. What are we elongating? What are we making longer? Yep, we're making that strand of polypeptides longer. So there's a hint. Anybody, one last time, we'll guess what going to be? Yes, perfect. So they're right next to each other. Again, it's hard for you guys to see it. I'm asking you to pretend like it's there. So now you show you a picture of it. Once it's there, now we're going to add it to it. Like, all right, well, there's the new amino acid. Let's add it to the chain. And that's what happens. That's step 2. Peptide bond formation. Because remember, I don't know why, but some biologists decided to name that particular bond. So even though it's a covalent bond, just like any other, we call it a peptide bond. But anyway, yeah, just like Levdi guessed, we're having that bond, right? We're elongating that strand of polypeptides. It just so happens, and I'm not going to ask you this too, but if you notice in the picture, the new amino acid, and it's actually kind of hard to see, the new amino acid is not added to the strand. Technically, the strand is added to the new amino acid. As you can see, this little arrow goes from left to right. It's hard to see, but that's what happens. I'm certainly not going to ask you that. Yeah, what you really need to know for the exam, again, in step 2 is when the peptide bond is forming. That's basically it. All these other details, who cares? But yeah, technically what happens, and you can't see it in this picture, but after this has been attached to that, this tRNA on the left is useless, right? It doesn't have any amino acids. It's done its job. So what they're not shown here is that thing just leaves, and then the one on the right shifts to the one on the left, and that's what this next picture is. The tRNA is out of there, right? That tRNA on the left, it did its job, so it leaves. And then what happens is, and you can barely see it in the picture, but this would be better with an animation. Again, imagine this tRNA that's on that right slot scooting over to the one on the left slot, right? So the one, it's taking the place of the one that just left. And then therefore you'll have that spot open again on the right, and the whole process continues. So yeah, this is all part 2, right? So when we're talking about translation, initiation, elongation, termination, right? And we're talking about elongation, and elongation itself is broken down into 3 steps: codon recognition, peptide bond formation, and translocation. And hopefully that's easy for you to memorize, because when you think about it, again, this is one of those, I love it because it's one of these very intuitive things, right? It just has to happen that way, right? First, you're going to have something recognize the codon and come in with its amino acid. Next, you're going to have that amino acid attached to the other amino acids. Next, you're going to need to move everything so you can start the process again. So it's all very intuitive. So the question is, you know what I forgot? What I did not mention? Oh man, I need to fix this. So let me back it up really quickly. I'm sorry. Like we said, the first step is called initiation, right? Initiation. Oh no, I did bring it up. Never mind. Okay. So initiation brings all the parts together. And technically, even though it was on the slide, I'm not sure I said it. So let me just say it, just to remind you. When that tRNA first binds to the mRNA, that spot is called the start codon, because that will be a test question. So just like in initiation, I'll say, where does initiation of transcription happen? Remember, that's the promoter. So then the other question, where does initiation of translation happen? Start codon, yes. So they're different, different words for initiation, transcription versus translation. But the good news is when it comes to termination, it's all the same. Well, as far as nomenclature is concerned, it's all the same. Oh, no, that's not true. It's not all the same. There are some differences. I forget that. Anyway, termination, right? So this is where all the processes end. Where does it happen? It happens at the stop codon. So yeah, I got ahead of myself. So remember, for transcription, it stops at the terminator. Here, it stops at the stop codon. Just so you know, and I'm not going to ask you this, but there's 3 words that say stop, right? If it's a UAA, a UAG, or a UGA, that's those, all 3 of those words say stop what we're doing. The process is done. So neither one of those 3 codes for a specific amino acid. It just says stop translation. That's the thing that says we're done. And then all the parts just go away. The polypeptide is freed. The tRNAs go and do their own thing. The ribosome splits up into different subunits again. And that's what happens. So then the question is for independent work, if you want to look into it. So what happens, right? So, okay, let's talk about the ribosomes for a second. Like I said, the small subunits split up and they go their own way. Is that ribosome able to be reused again? Or is it done? Is it like a one-use thing, or are ribosomes reusable? Same thing with mRNA. What happens to that? We just read it. We used it to, we read the code, we built the polypeptide, and now all the parts are just going off and doing their own thing. So what happens to that mRNA? Does it get used again to make another identical strand of protein? Is there something in there that destroys it in the cells? This is all for you to look at. I'm not going to ask you. They're all for independent work. You can look into it. Same thing with the tRNA, right? So the tRNA has been used. It no longer has its amino acid. So is it going to be used again? Is there something else that says, all right, well, let's reload you with your amino acid? So all things you can look into. But anyway, back to this discussion, back to the exam. The only thing you need to know about termination is this is where the process stops. It stops at a particular stop codon. That's the name of the thing that happens. And at that point, everything goes on its own way. Another thing I didn't mention, sorry, before I move forward, is also the polypeptide itself is not done. We don't talk about what it does in this class, but it does. It has to go through some changes too. It's not ready to go. Right now, it's just a polypeptide. Remember when we talked about this a long time ago? Technically, it's not a protein until it's folded into its particular shape. So those are other things you can look at. What does that? What folds the protein? What processes the protein? And actually, you could look at the chapter 4. Remember, we kind of skipped chapter 4, or whatever cell it was that talked about how cells work. Yeah, you can look that up there if you want, but I'm not going to ask you on the exam. So any questions about the whole process in general before I go through a really quick review to talk about everything from DNA to RNA to protein? All right, then, here we go. From a big review really quickly. And again, I will go through this quickly because this is not new information. Now that I've told you all the specific details, we're going to go back and look at it again in the big picture. So again, this flow of information goes from DNA to RNA to protein. Memorize that before you memorize anything else. Then once you have that memorized, memorize the fact that going from DNA to RNA is called transcription. Right? You're going from RNA to protein is called translation. Then recognize transcription happens in the nucleus. Translation does not happen in the nucleus. Then memorize the fact that transcription, right after you've made that molecule of RNA, it also has to go through some processes, right? It has to be changed before it can leave the nucleus. And we talked about that, right? Cap and tail and alternative or splicing. Sometimes alternative splicing. All right. This also points out, okay, this might be new information, but I'm not going to ask you. Translation, you have one ribosome making an average size polypeptide, and it does it in less than a minute. So you don't need to know that, but there you go. And again, something you could look up for independent work. Like specifically, what's the number? And I'm sure it'll be different depending on your source, because this just says average size polypeptide. Well, what's an average size polypeptide? Now think about how much some of them are much larger or smaller. Another thing you can look into that I haven't mentioned, what about quality control? What happens if something says, all right, I need this specific amino acid, and then the wrong tRNA comes in and like, yep, here you go, and brings the wrong amino acid? Does that ever happen? Is there anything to fix it if it does happen? Does the mistake just get unchecked if it does happen? All those things you could look into. But again, DNA to RNA transcription, RNA to protein translation, and then of course that polypeptide also needs to be processed. Here's a picture of what we're talking about. There we have the DNA inside the nucleus where it's safe. That's our master copy. This RNA polymerase comes in and reads it, right? It reads the code. It opens it up so it can read it. It starts at the promoter. It works its way down to the terminator. It's making the strand of mRNA one nucleotide at a time. It gets longer and longer and longer. Then that mRNA is done, but it needs to be processed. So we add a cap, we add a tail, we splice out the introns. Now that mRNA is ready to leave the nucleus. Here's a picture I haven't shown you, but I did bring it up, right? And I said you could look this up for independent work, and you still can because I didn't give you any words here, any names. Here on the side, all right, so we're paused. We're not looking at the mRNA for a second. Right now we're like, all right, so that's what happened with the mRNA. And meanwhile, in the outside of the nucleus, we have this tRNA being made, right? So we have this tRNA that's being attached to its specific amino acid. There's an enzyme doing it, and it's requiring energy. But again, I didn't tell you the name of the enzyme, so you can look that up for independent work. And again, is it like a one-size-fits-all enzyme, or is it one specific enzyme for each specific tRNA? Things you could look into. Anyway, so now we have our tRNA ready to go here. We have our mRNA ready to go there. So the mRNA leaves the nucleus. And then now we start translation, right? All the parts come together. The small subunit of the ribosome, the large subunit of the ribosome, the mRNA, that first tRNA with its MET, right? It comes in, we're going through elongation, so we're making it longer and longer and longer until finally we hit the stop codon that says, all right, we're done. And then all the pieces go their separate ways, right? The tRNA, the empty tRNA goes away. The subunits separate from each other. The mRNA goes its own way. That polypeptide is then, it's not showing it, but it goes somewhere and it gets processed and changed into the actual protein that we're after. Whew. So, any questions about that picture? Again, this is just a review. So, transcription and translation together is how genes control the structures and activities of cells. Just to remind you, big picture stuff here, right? So again, you know DNA to RNA to protein, but just to remind you, this is why it's so important. So even though we're only building proteins, just remember, everything that's happening inside your cell is happening because of specific enzymes. And, those are made of proteins, right? So we're building these little machines, these little enzymes to do jobs. So that's how genes do this, right? So we need the cell to be doing this job. So the cell's like, okay, well, then we need that enzyme that does that job. Therefore, we're going to need to read the code for it, right? We need to read the gene, make the mRNA, and then use that to make that protein. Yeah. Anyway, any questions about this slide? Okay, so I like that your textbook points this out too, or at least I think so. I know the old textbook did. So here it is. This idea of going from DNA to RNA to protein used to be thought to be the only way that genetic information controls traits, but there's been recent discoveries, and RNA has so many other uses. This 100 level class for non-biology majors, so we won't get into it. But for you to look at chapter 11, which we're skipping, and look into all these different roles of all these different types of RNA, which is a good time. Yeah, so that's a good time to remind you, we are skipping chapter 11. So if you're following along, you don't have to do that. Anyway, any questions about this slide? Okay. The perfect time to stop then. When we get back, we will talk about mutations. Yeah, and that's it. So I'll be done. I'm going to go to my office and start checking your labs. So if you've already turned a lab in, I'm committed to looking at it. Then hopefully you won't have to come to lab today. Any questions about anything? All right, then I guess the last word for attendance will be agents. You can just see it at the bottom there. Agents is the last word for attendance. Other than, again, ask me one question about the material, and that's it. I'll see you either on Friday or in lab.